Hello everybody and welcome back to the iCow soil biology class. This is the second lesson and today we're going to be talking about bacteria, more to the point, soil bacteria. We'll start by reminding ourselves about the soil food web, which shows us who eats who in our soils, who's dependent on who, and how collectively all the creatures in the soil help to stabilize the soil, regenerate it, keep it healthy so that we can grow healthy crops. The soil food web is divided into trophic levels, with the first being the photosynthesizers. The second trophic level are the decomposing mutualists, pathogens, parasites, and root feeders. You can see that down there on the left side of your screen at the bottom. Bacteria are part of this group, second trophic level. They're very, very, very tiny, microscopic in fact, but extremely numerous. One small bacterium may seem in fact insignificant compared to any other life form on Earth. One bacteria versus, say, one elephant. However, bacterial biomass is more than the biomass of all the organisms in any ecosystem. By the way, did you know you have a few trillion bacteria in your own body? New discoveries about what is inside the body are making scientists rethink what makes a person human and what makes people sick or healthy. Less than half of the cells in the body are human. The rest belong to microorganisms that affect the health, mood, and whether certain people respond better to certain medications. So to our 30 trillion human cells, we have on average about 39 trillion microbial cells. So by that measure, we're actually only about 43% human. Whenever we hear about bacteria, though, we hear bad things. We hear about bacterial infections of people and bacterial diseases of plants and animals, and we mostly associate bacteria as being bad. But did you know that there are good bacteria and there are bad bacteria? And we call the good ones beneficial bacteria, and the bad ones generally are disease-causing bacteria. But we need both of them to exist in the world, and we need a healthy balance of both. And healthy systems do prefer good bacteria. But if we didn't have the bad ones, we'd have a lot of stuff rotting on the planet and nothing to decompose it and make its nutrients ready to be recycled back into the circle of life. We just need to have a lot more good bacteria around to compete with, consume, and inhibit the bad bacteria from having negative consequences on our health and the health of our crops and livestock. But I guess to a decomposing body, bad bacteria are good. To our body, where they will affect us, we see them as bad. So we tend to term bad bacteria bad in relation to how they affect us and the things around us. When we think of bacteria, we know we can list the bad ones that affect humans, like cholera, E. coli, salmonella, but we can start to list the good ones, those that are in our gut, bifidobacterium and lactobacillus, that help us to have a healthy gut. The bacteria in our gut are called our gut microbiome, and the same in our soils are called the soil microbiome. Bacteria come in quite a few different shapes and sizes, and some of these help us determine whether they're beneficial or disease-causing. As we're looking through microscopes and looking at these different shapes, we can pinpoint which ones generally are good and which ones are disease-causing. Beneficial bacteria are usually round or rod-shaped. They can be single or joined up in different formations. A single round bacterium is called a cocci. Two together are diplococci. A string of round bacteria are called streptococci. The rod-shaped ones are called bacillus. They too can be found as a pair or in chains. When we look at bacteria under the microscopes, the shapes help us to determine where they fit, good or bad. Some can move and some move extremely quickly. We call this being motile. They do this by using a little tiny string or hair-like whip on their bodies called a flagella. Some have more than one flagella. Some have many, but they're so tiny that they can only be seen under very powerful electron microscopes. These little flagella whip around and give the bacteria motion, sometimes forward, sometimes sideways, up and down, all over. Most of the disease-causing bacteria are motile. They move really fast. And if left unchecked, they can create serious illness and disease and even death. The three main disease-causing bacterium in the soil that we look out for under the microscope are Vibrio, Spirilla, and spirochetes. Spirilla rotate quickly like a corkscrew and move very fast. Vibrio flip around like a comma on steroids, and spirochetes move in a swimming fashion like a snake in water. We're told that if we see these, we must wash our hands, handle samples with care, and dispose of them very carefully. We don't want to see these bacteria in our soils. If we have an abundance of beneficial bacteria, then actually we shouldn't. There are some pictures of the Vibrio, the Spirilla, and the spirochetes at the bottom there. Like I said, they're very fast moving. Beneficial bacteria do wonderful things in our soils. They suppress disease by keeping the disease-causing bacteria in check. They build soil structure through their production of glues, forming microaggregates. They help retain water and nutrients in the soil. They produce energy-efficient decomposing enzymes that help them strip nutrients from parent soil material. They hold many materials in their body, and these are then released to plants as plant-available nutrients when they're eaten, digested, and pooped out by other soil organisms. They're also really good at decomposing and neutralizing toxins. Some bacteria can digest oils, and they're being used to help manage oil spills in the oceans. And finally, good bacteria help the soils maintain an alkaline pH, which is favorable to most crops. Here's the thing about bacteria, though. Here's the thing about beneficial bacteria, though. They only survive in aerobic conditions. This means there must be oxygen present in their surroundings. Makes a lot of sense for plants too, as they too must have access to air and oxygen in their root zones in the rhizosphere. Disease-causing bacteria, as the name implies, are not so good to have in our soils, but they're also really good energy-efficient decomposers, producing enzymes suited to their condition to do so, and to their roles. Unfortunately, they also produce toxins, they cause disease, and they lower the soil pH, making it more acidic, which most plants don't like. Think of a dead animal decomposing. If we didn't have anaerobic bacteria, they wouldn't decompose properly. So they have a use, but in the soils, their use for our crop production systems is fairly limited, and we like to make sure that the beneficial bacteria rule in our soils. The next slide shows us the difference between aerobic and anaerobic microbes. Aerobic ones, remember, these are the good guys. They like conditions where oxygen is 6 milligrams per gram or more, and the anaerobic ones like it when oxygen levels are below 4 micrograms. So those are the aerobic ones, and then we've got the anaerobic ones down here. You can also see that lower oxygen levels lead to more acidity. And then there are a few bacteria that I haven't introduced to you yet, and they're the facultative bacteria. They live in the zone between aerobic and anaerobic, and to do so, they have to produce a double set of enzymes to do their work. Enzymes that work in aerobic conditions and those that work out of anaerobic conditions, and they have to switch between the two of them. As such, they're the least efficient of the bacteria. However, when we see these guys under the microscope, they're great indicators of systems that are going anaerobic, and they can help us make decisions to reverse this trend before we add soil amendments like compost teas to our fields that might be anaerobic, and in which case they're not preferred by our crops, and we would be able to react and do things about this if we were able to see this. You're probably wondering what happens to the good guys when conditions change, or even vice versa. What happens to the bad guys when conditions change from anaerobic to aerobic, right? Well, let's look at how those changes might occur to begin with. Changes in the environment result in changes of types of bacteria. Bacteria types in the soil change when amounts of other things, including oxygen levels, change in the soil. But what causes oxygen levels in the soils to change? The three main causes are compaction, water stagnation, and suppression or removal of soil microbes, including bacteria. So, Let's look at compaction. This is a term used to describe very hard soils. Soils that are so hard that roots can't penetrate through them. Soils that we believe need to be tilled to turn and to aerate. It can even refer to a hard pan under the soil. What it means, though, is that the soil particles have collapsed upon themselves and there's very little air in the soil. Think about how this affects aerobic bacteria. Where there are no airways due to reduced soil microbes, beneficial soil microbes can't exist because they need oxygen. Compaction changes oxygen levels in the soil and thus leads to successful environments for who? For the anaerobic disease-causing bacteria to exist. When we till our soils, we do so to remove compaction, but are we really doing that? Or are we disrupting the soil habitat for the aerobic microbes, putting them into a position of flux where they either die, go into dormancy, or move, and leaving the soils in a state where they become compact? When we're doing that, we're positioning our soils, we are physically and almost purposefully putting our soils into a position where they're likely to become compact, therefore anaerobic microbes are going to love them. Let's look at water stagnation. And why does water stagnation happen? When it does happen, the airways get filled with water, and there's therefore no more airways in the soils. This creates the soils to collapse, and it leads to compaction again. But why does that happen? And it normally happens when you don't have good soil aggregation. So there's not enough life in there creating those glues and creating that good soil integrity. And those microaggregations. But who causes those? Who builds microaggregates? And it's the beneficial soil bacteria. So again, compaction is a sign that they're not there. Water stagnation results basically in reduced oxygen levels in the soils, and once again causes the perfect environment for disease-causing bacteria. And finally, let's look at suppression or removal of soil microbes through chemical or mechanical means. Tillage and the use of all of the sides, the pesticides, herbicides, bactericides, etc., all lead to compact soils and the perfect environment for disease-causing bacteria. Because they do what? They upset the environment for the aerobic microbes. And when there are no aerobic microbes, soils collapse, aggregations are not built, soils collapse, soils can basically end up being eroded, but it gives rise to those disease-causing bacteria, which we don't really want to have. So let's have a look at this graph again. At any one point in time, soils have aerobic, facultative, and aerobic bacteria in them. It's not as if these bacteria come and go anywhere. They're there, and something happens to them when conditions change. And oftentimes when that oxygen level changes, it has the biggest influence on what happens to these microbes. And we ask ourselves then, what happens? What happens to beneficial bacteria when the situation does become anaerobic? Or what happens to anaerobic bacteria when the situation becomes aerobic, when there are sudden changes that change that? So if the change is very sudden, like really, really, really sudden, like you've gone and sprayed on a whole bunch of chemicals, or there's been a big flood, or there's an earthquake, or somebody's come in and tilled the land. If there's a big change, many of them die. If the change is moderate, but still it can be quite quick because they can adapt quite quickly. This is the coolest thing about them. They go into dormancy. They produce a whole bunch of protective glues around their body. They turn into little spores, and those spores have a substance around them that protects them from anything harming them, like a little shell around them, and they sit in there and they wait. Bacteria, both anaerobic and aerobic, can stay in these dormant stages for thousands thousands upon thousands of years. During the change, the bacteria is suited, during any change, the bacteria that is suited to the prevailing or unfolding condition simply come out of dormancy. So for optimum soil health, we should aim to preserve and create conditions for beneficial bacteria. We'll keep the bad bacteria at bay. We'll produce wonderful aerobic conditions in our rhizosphere, that's our root zone, so that our plants can actually benefit and grow healthy, and we then don't have issues with pests and diseases. So think about your farming practices. Do you support or do you destroy beneficial bacteria? Here's your homework. Get a piece of paper and write down 5 ways that you've been helping your beneficial bacteria. Now, up until this point in time, you may not have thought that you have any practices that do, but there are some that do. Practices that don't disrupt the soils, practices that don't create compaction, practices that keep the soils aerated and stabilized. Do you follow any of those practices? Because those are those that help your beneficial bacteria. What about 5 ways that you've been damaging your beneficial bacteria? So again, on that same piece of paper, write down 5 ways that you've been damaging your beneficial bacteria. We've learned that plowing damages bacteria. We've learned that spraying using toxins damages beneficial bacteria, and even flooding. Some of us use flooding systems for irrigating our crops, and in the process we run the risk of creating an anaerobic environment, which does not support aerobic bacteria. We may do that in our compost piles. We may produce compost and cover them up so tightly under the hot sun, wet them, damp them, keep them covered. Underneath there you're brewing anaerobic microorganisms because they can't get any air. They will break down your compost, but eventually if you bring that compost and put it onto your crops, put it into the root zone of your crops, you're suddenly exposing the roots of your crops to a whole bunch of disease-causing pathogens and bacteria, and you've made them yourself. So making compost the right way, making sure that your compost is being made with a focus on it being aerobic, you're turning it, you're not covering it and suppressing any oxygen or air flows within your compost. And then think of the 3 things that you'll do now going forward to preserve and create conditions for beneficial bacteria to thrive. I hope that this lesson has helped you to think a little bit differently, to think a little bit more about how we can try to manage and use our bacteria and soil biology to help us farm more constructively and in line with healthy systems, and also without all the backbreaking work. Let the biology do the work for us. If we don't need to till, let the biology do the work for us. Thank you for being here. I look forward to seeing you in lesson 3 next week, which we shall be discussing part 2 of bacteria. Bye for now.